When people think about inferential logicians, they think about PAC and learning and all that, which is just not there. But there's this other thing called statistics, which has a broad collection of ideas. And one of the broadest is the selective inference problem and the family-wise error rate problem. It's one of the big problems of statistics, and some of the best, most important progress has been made over the last 2 decades in this particular topic. And one of the leaders, if not the leader in that topic, is Yoav Benjamini. And I'm just going to, without any further ado, turn it over to him and get him his full time.
Thank you. Thank you for the opportunity to speak at this occasion, the opportunity to return to Berkeley for a visit, which I do often. Let me start, but 2 apologies. First of all, this apostrophe shouldn't be there. So that's one thing. The second thing is that my hearing is not good, so when you ask me something, please speak up. Okay? So even though I have all the right aids, I don't hear you well. And the third thing, did you distribute the syllabus for this talk?
No.
No. That's good, because I am not committed to anything that I have to talk about, and I prepared a lot, but if I'm not covering it, that's fine. That is, we'll do it at the right pace, and don't be afraid to ask, to stop, and so on. If I feel that it helps us, I'll tell you. Okay. So I'm going to speak about, first of all, about the whole idea of simultaneous and selective inference, and then talk about testing with false discovery rate. This is sort of the more basic part, and that will be about the first half of the talk, and then I'll talk about false coverage rate estimation and the connection to model selection ideas and to more complex current research problems. And actually, I would like to start with what I think is the first data mining problem in statistics, and maybe even in science at large. And this is the kind of experiment reported by Steele and Torrey in the 1960s, and they go back to the work of Erdmann, who is an agriculture researcher. And he was, according to them, he was dealing with 6 groups of red clover plants, each inoculated with a different strain of Rhizobium bacteria, just in order to increase the nitrogen content. And there were 5 measurements of nitrogen content on each group. And we can certainly model it by the average, the estimator in the group being normally distributed with some mean mu and standard error of sigma square over 5 because of the 5 measurements, and we have 6 groups. The reason I bring this here is that this is the most standard example of the kind of problem that I'm going to show you, and it is quoted and it is brought up in SAS book Explain the Material and BMDP and many textbooks and so on. It's really a sort of a standard example in this setting. What are we interested in this case? Well, you want to know the differences between the groups. So you can estimate the difference by comparing the average or the mean, the average in one group minus the average in another group. Then you want to know whether it is statistically significant. So you set up the null hypothesis that there is no difference between the parameters, and you can do a 2-sample t-test or a 2-sample z-test if you know the sigma squared, and so on, in order to decide whether this difference is above what's expected under the noise in the data, and so you can reject the null hypothesis in this case. Or you can do it by a calculation of a p-value. That is, you calculate what is the probability, if this is in this the case, that the standard random variable will be bigger than this difference, appropriately standardized, that this is supposed under the null hypothesis to be normal 0, 1. You calculate this one, and this is the p-value. And if the null hypothesis is true, the p-value has to have a uniform distribution over 0, 1. So once you have calculated the p-value, in order to reject HF as you can reject H naught, and you will do it in producing a type 1 error only with probability less or equal to alpha. So this is the standard setting of hypothesis testing, either in this form or in the sense of the p-value form. Okay. in the language of this talk, we will say that we will make a discovery with the probability to make a false discovery, that is, the null hypothesis is true, but we are deciding on that the difference exists with a probability less or equal to alpha. And we'll reject if the p-value is small enough. That's the regular things. Now, why do I call it the first data mining problem in statistics? Because in this kind of problem, as I described, we might select the most promising group's difference. That is, we have k groups, we'll take the largest average minus the smallest average, and we'll look whether these two are statistically significant, whether these are above the noise variation. But with 6 times 5 over 2 different possible pairs that I can compare, then even if I do each one at level alpha, if I take the probability of the largest minus the smallest and divide it by the probability thing, that will clearly be less than alpha. So even if the null hypothesis is true, there is no difference between those groups whatsoever, just taking the largest minus the smallest will give me something which is biased. Too often will I get a false discovery. And of course, the larger k, the more groups there are, the worse the situation is. Well, indeed, when going back, we tried and were able to find the original paper by the agricultural researcher, and he had 13 groups. So we had 13 times 12 divided by 2, 78 pairwise comparison. And with the limiting power of the 50s, actually of the 40s, this was a data mining problem. That is, you had to calculate only 13 numbers. But the inference problem became huge. And suddenly, questions like, do I keep what I do on a single comparison? Do they keep the properties when I'm moving to a large problem? Became relevant, even at that age. So that's why I call it the first data mining problem in science. And this is really the origin of the multiple comparison problem area, or as it's called, MCP, multiple comparison problem. And in fact, the area flourished during the 50s, making some of the basic ideas that I'll present in the first few slides are from that era. Now, let me demonstrate just what kind of problem do we have with this kind of phenomenon. This is taken from the significant thing. So scientists study rumors. Eating colored jelly beans causes acne. Okay? So there was a study of purple jelly beans, and in fact, they don't seem to cause acne because the p-value is bigger than 0.05. And scientists studied where the red ones are, and in fact, they do not, because it is bigger than 0.05, no significant evidence. And so on, and jelly, and then comes the green ones. Oh, we found a link between green jelly beans and acne. The p-value is less than 0.05. Wow. Okay, let's continue, and continue, and continue, and finish our study, and there comes the news. Green jelly beans linked to acne, 95% confidence. There is only 5% chance of coincidence. This is the essence of selective inference. You select out of the many things that you looked at, you select something that looks promising, interesting, something like that. Okay, that's a cartoon, but it's not only jelly beans. This is from a study, data mining petition at Kaggle. Unusual secrets are hidden in numbers. For instance, an orange car is less likely to have serious damages that are discovered only after purchase. That's exactly the same study with exactly the same phenomena, but here it is. Okay? So we are still, by the way, there is an explanation for that, of course. Okay? Because those are very unusual cars, and people keep good track of their, they take good care of them and keep them up in garages and give them usual service and so on and so forth. So there was also an explanation of why this happens. But it's not only colors and jelly beans and so on. Giovanni and others in the 95 examined the possible effects of excess eating of 130 different kinds of foods on prostate cancer. And there were 3 kinds of food that cleared the statistical significance bar. and these are the only ones reported in the article abstract. And the conclusion was eat ketchup and pizza to prevent prostate cancer. And the reason I encountered this study is that I came to visit my friend, and she is a very food-conscious person, and she offered me a pizza for the first time in my life. And I said, "What's that, Rita?" And she said, "Oh, it's good for your prostate cancer." And that's how I found about this one. Now, unfortunately, in the article itself, all 130 results are there. I mean, there's no hiding, there's no mischief in that, everything is reported. But the abstract carries this information only, and the abstract information, the abstract is the selection process by which the interesting findings, the significant findings are brought in, are passed to the public, and in fact, even to professionals. I mean, ask your colleagues or friends who are physicians whether they read the paper or they read the abstracts. Okay, the answer I think is clear. Now, just for curiosity, I checked a year ago what situation with this kind of study. There were it's cited over 1,000 times, dozens of studies about the contribution of tomatoes to the healing of different types of cancers with unclear results. Unclear results. You have results in this direction, in the other direction. The most recent study that I've seen at that time claims that the secret is in the oregano. And it was published in TV before it was even presented in a conference. So that's the nature of significant findings. So what is our first line of protections against this phenomena? Well, since the danger seems largest when all of the difference, I'm going back to the original question of this pairwise comparison. Since the question seems the most severe when all the mu's are the same, because then you have the most possibilities to have a type 1 error, then test this hypothesis that they are all the same, and you can do it via F-test at level alpha. Those of you who don't know what an F-test doesn't matter, but it tests just this single hypothesis. And if it's not significant, stop. That's the first line of protection. But if it's significant, continue and do all pairwise comparison as we did before. This is called, actually it's a suggestion by Fisher. It's called the Fisher, Tukey gave it the name Fisher protected LSD. Not because of the material, because it treated it as the least significant difference that one can think of. Now, why is it the least significant difference? Can you see where's the problem with this first line of protection? Well, suppose one of the mu's is far away from the rest of them. You are about to reject this first line of protection. Now you have with the problem which just instead of k things in the same place, k minus 1 things in the same place. Just a very little bit less of a problem, but mostly everything is just the same. So if we want to, so this kind of protection was given the name Families error rate in the weak sense. That is, if you manage to control just this probability, this is called the family-wise error rate in the weak sense. That is, that the null hypothesis that everything is equal, the probability of making even one type 1 error is less or equal to alpha. Now, at this point, I need some notation in order to continue. We are talking at the problem right now about testing many hypotheses. So you have H1 to HM. M naught of the M hypotheses tested are true, meaning that we have only M minus M naught, which are false, meaning M minus M naught potential, real potential discoveries. All the rest are, if we discover them, are in error. Now, we do not know which ones are the true ones or the false one. We don't know their numbers, and at some point of time, though, we'll try to estimate this number at some point of time. But generally, we just assume that the result of any testing procedure is Ri. Ri equals 1 if Hi is rejected, if a discovery was made, 0 otherwise. And with Vi, we measure whether an error was made, whether we rejected, but Hi is true, that is, we made a type 1 error or we made a false discovery. and it is defined as 0 otherwise. Overall, we look at all the number of the hypotheses rejected out of the M, that will be R, and the number of hypotheses rejected in error is V. So the weak family-wise error rate that we defined before is really the probability that the number of errors will be greater or bigger than 1. So that's just a formulation of what we said so far. Now, the second line of protection, okay, since this is not enough, we want to protect against any configuration. The mu's can be one far away and all the rest together. They can be two groups, and in each one m over 2 things mu's together. We want to protect against any possible configuration and not to make even one error when we make inference, the many inference. So the family-wise error rate is the probability of making one or more errors in our study. So by having a method that assures that the family-wise error rate is less or equal to alpha, the probability of making even one type 1 error in the family is controlled at level alpha. And this is also has the terminology of being called the simultaneous inference. That is, all inferences we're going to make about all the M hypotheses are going to hold jointly. We're not allowing even one error in our body of inferences. The same is true when we are trying to put confidence interval. Confidence interval, we're trying to put, we have a quantity like estimating the difference, and we want to put a confidence interval that states that, with a high probability of, say, 0.95, it will cover the true unknown value of the parameter. That might give us information about the size. You can similarly define V to be the number of intervals failing to cover their respective parameter. And if you have, for any configuration of the parameters, the probability that even one of them will fail to cover less or equal to alpha, the set of such interval is called to have simultaneous coverage, level 1 minus alpha. Okay, this is clearly a protection. I mean, we've got it nice. And I will take the bother and discuss the old and trusted solutions, because some of them are so oddly presented that it worth emphasis. So what happens if we test each hypothesis separately at level alpha bon, some level alpha? Okay? And I look at the expected number of errors. The expected number of errors that I make is the expected of the sum of the VI, each one measuring an error in a particular hypothesis. The sum of expectation of the sum is the sum of the expectation. And this is less or equal to m naught times the level at which I'm doing each test, because VI is a 0-1 random variable. Okay? M naught is the number of true null hypotheses. It's certainly less or equal to the number of hypotheses I test. So this one is m times alpha bon. So if we want to assure that the expected number of errors that we make is less or equal to alpha, we may use alpha Bonferroni, which is alpha over m, and this is the famous Bonferroni method. Did we need any condition here? No condition on the distribution, nothing at all. Unfortunately, the Bonferroni is often referred to as something that holds only under independence, and that appears in textbooks. So that's why I'm making here an effort to convince you that it is a simple property, hardly any inequality involved. It is just the expectation of the sum is the sum of the expectation, and that's it. And so we have that it controls under any configuration the expected number of errors is less or equal to alpha. Well, I didn't talk about the expected number of errors. I talked about the probability of making even one error. Before that, the family-wise error rate. Well, protecting this way, it also assures that the probability that V will be bigger or equal to 1 at level alpha. And that's because the simple thing that the expectation of V is 0 times the probability that number of errors will be 0, plus 1 times the probability that it will be 1, plus 2 times that you will have 2 errors, and so on and so forth. But we can always reduce it. This one will be 0, we reduce this one to 1, reduce this one to 1, reduce this one with 1, and we have the probability that V is bigger or equal to 1. So if we use alpha Bonferroni for each individual test, or for each confidence interval, we make it a level alpha over m, the family-wise error rate is at less or equal than the expected number of errors, and therefore less or equal to alpha. Okay? This is sort of the first method that was invented. Now, there was no condition here. Can we gain if we introduce some strong condition? For example, something about the distribution of the test statistics. So we can say that what happens if the test statistics are independent? Because if they're independent, I can actually calculate probabilities. So if the test statistics are independent and we test each hypothesis separately at some level alpha SID, then the probability of making even one error is 1 minus the probability of making no error. And this is 1 minus alpha SID to the power of m naught, sorry, which is less or equal to raising it to the power of m, which is less or equal to alpha. So if we want this one, we simply have to take the alpha level for each test at 1 minus alpha to the power of 1 minus m. And this is Sidak's multiple testing procedures. And note that if m not equals m, then we have here equalities all the way, so in some sense it is the best that you can gain. Okay, so we use the dependency structure in this case to get a better test. How much better? Surprisingly, not that much. That is, alpha SID is 1 to the power of 1 minus alpha 1 over m, which is approximately 1 minus alpha over m with the squared Taylor. And so we have alpha Bonferroni, alpha over m, plus something which is the order of alpha square and divided by m times m minus 1. So the larger m is really the advantage will disappear. Even for small m, okay, there is very little gain. If we do the Bonferroni, we do 0.05. If we use the Sidak, 0.00511. Okay, so this is the amount that we gain. Some situation, like the one I started with, the pairwise comparison, we can gain more from utilizing the fact. I mean, remember, if you do group 1 minus group 2 minus group 3, there is very strong correlation between the two. Correlation is positive. In this case will be negative. Group 1 minus group 2, group 1 minus group 3, the correlation will be positive. Okay? Tukey's procedure for pairwise comparison utilized the dependency by calculating the distribution of the maximum of this one. That is the largest minus the smallest average when they're all normally and independent, but once you take the differences, there is this structure. It's the same idea as was here, but a larger gain. And interestingly, this is known as a yeah.
I have a question. So if you use this method, the Bonferroni or the
Bonferroni?
one, will it affect the number of type 2 errors you may have?
The effect of?
The number of type 2 errors, because we're just talking about type 1 errors,
Yeah.
right? So if I reduce, I replace my alpha with alpha over m, does that affect the
type 2, I mean the
type-
probability of making a finding.
Yes.
Yeah, the power, obviously.
Okay.
Obviously. That's why there is a strong balance and the hesitation of many people to admit that they have this problem because it harms the power tremendously. And I'll demonstrate this point.
Okay.
I'll demonstrate this point. Okay, so this is known as post-hoc analysis. Again, this is another name that is given to data mining. Data mining is post-hoc analysis of your data. The point is that you look at the data and then you select, in view of the data, what to infer upon. That's what the point is in all those examples. Last one in this point, newer solution. Well, newer should receive some qualifications from 79, but it's much newer than the other ones, and I will use it later on. If we look at the p-values, each p-value for each hypothesis, and we order the p-values for smallest to the largest. So this is the smallest one, second smallest, and so on. This is the largest p-value of the m of them. Now we compare p1 to alpha over m, a Bonferroni type of thing. If it passes That is, if it is rejected, we look at the second one, but compare it to alpha over m minus 1. The idea, I mean, we are sure that 1 is okay, so now we have to protect against m minus 1 hypothesis, so we can do it with alpha m minus 1. And so on, we continue until for the first time we cannot reject and we have to stop at this point, even if later on there are some things like that. And it's interesting to know that it always controlled the family-wide zero rate. In spite of this sort of greedy method that we are doing it, it always controlled the family-wide zero. Again, no strings attached. No distributional assumption, nothing at all. The only problem, you can't make confidence interval with this kind of thing. That's, okay. Let me give you an example from my own research. So, I've been involved over the years with research with mice behavior and genetics of mice behavior and so on and so forth. And what I will show you now is the film of a mouse in our laboratory exploring a new arena. Oops, sorry. So what you see here is the mouse getting out of the hole and exploring and going back to the hole. Goes out, notice that he makes stops and then moves again, then he goes back to the origin. He goes a little bit away from the wall, goes in the other direction, makes stops, another stops, goes back here. Then comes another excursion outside into the wildwood. He will go all the way here, make another one here, and go back to the hole. And this kind of extremely structured behavior, we managed to measure it, to quantify it, and really to study its growth and so on, because it will go on like that, and it may go on like that and return back home. And then suddenly he will realize that it's the same thing, and he will make a few rounds like that very happily, running around the arena, and then he will start to explore the center. Okay. What I'm going to use this example is to demonstrate the use of the p-values and different methods. And this is the result of a large phenotyping effort done in 3 laboratories at Tel Aviv University, NIDA, and MPRC. Okay, so you have here different behavioral endpoints like the number of progression segments each time that he gets out of here, the time away from the wall, distance traveled, the number of excursions that you have seen. With all kind of quantification of the behavior, in this case 17 different measures. We compare them across laboratories with a mixed model, and here are the p-values, and they are already sorted from the smallest p-value to the largest p-value. And if I use an unadjusted, okay, if I use the regular thing, I would declare all of these to be statistically significant. I have a lot of findings in terms of the difference. Oh, this is done between 8 strains of mice, and so I want to know, is there a genetic factor in this kind of behavior? But if I take the Bonferroni one, all I get is the 0.002 line. Well, that's a big difference, and that's the point that you were making. A big difference between using the unadjusted one and protecting against the effect of selection in this form. And that's really the reason why many people try to avoid it until they have to. So, for instance, the FDA, drug approval requires very strict adherence to the multiplicity issue to protect you in some way against this kind of selective inference. What happens in this example? In the search for food affecting prostate cancer, these 3 food intakes that we're reducing, if you use the Bonferroni, they do not come out significant. If you use the Bonferroni, they do not come up significant. If you don't use the Bonferroni, well, those are the 3 that made themselves to the article. So again, there is an important difference between the 2. Now, with current research, the problem becomes more and more severe. I want to give you an example. I'll return to it at the end of my talk, if I get to it. If not, not. And this is from our recent research. trying to find association between brain volume in different parts of the brain and the genetic background. And this is based on the Alzheimer's disease neuroimaging initiative. And so the goal is to determine biological markers for Alzheimer's disease by testing for association between volume changes at voxel with genotype. So you bring the brains of the people that are scanned by MRI, you bring them to a common framework, and so you can check at each voxel volume pixel in the brain, that division of that, what will be the change, and associate it with a genetic marker, a SNP that can take two possible values and three possible values, AA, BB, and AB. This is the way that the original article reports it. You see this, these are 5 particular SNPs. For each one of them, the number of voxels, volume pixels in the brain that are searched are 32,000. What you see are just the few slices, but there are more slices. They are just not brought out. So there are 32,000 voxels searched. There is number of SNPs is 4,000, about 4,400, sorry, 440,000 SNPs along this dimension. Of course, you see only the first 5. So we are talking here about 13 billion tests of association done, and the original paper brings this one as an example. This is clearly a very strong selection from the pool of results that you have, and it is clear that it has to be adjusted for this strong selection. And in fact, it does. In neuroimaging, it is very customary to address this problem, usually to the problem of 32,000. This is really a different dimension, totally different dimension, and needs different methods. Now, what is the common point in large application that was demonstrated by these figures that I showed? In these large problems, the selected ones are presented, are highlighted, are discussed, the strength is displayed, the effect estimated, articulated, and so on and so forth. But those that are not selected are completely ignored. You don't know even what they were. They're not given name. In the good situation, like the 100 foods, they are somewhere in a table in the paper. Sometimes they are buried. In other examples, they are buried in the supplementary or in a database that if you wish, you can go and look at it. Sometimes even that is not. Simply, there is a clear distinction between those that are selected and those that are not selected. Okay? And not selected, I mean, they're just ignored. Now, my point is that increasing the scale changes the goal. And the best evidence that we have is from Tukey's own work. In 1978, remember, the Tukey's procedure is from the 50s. In 1978, in a discussion paper, Tukey wrote, one should always control the family-wise error rate. Okay, I mean, there's a clear statement that it's important in scientific and in other works to control this one. Well, in 1994, he was asked to consult on the National Assessment Educational Progress here in the States, comparing the results of the national test between the states. Now, 35 states participated in that, so the number of comparisons was 595. Now, even Tukey's correction is huge in this case. And this was a project at Nice, and there was a debate how to report the results. Do you report them as they come, or do you make some kind of adjustment to take care of the possible selection? And in fact, at that time, that was 94, Tukey was one of the first aware of our work on the false discovery rate, and the solution was to use the false discovery rate approach, which I will discuss in a minute. So it's the size of the problems and the loss of power that you mentioned that come with it that really made it necessary to try to, in some sense, to address the selection problem, but try not to lose as much power as it's done. So what about testing with FDR control? The false discovery rate criteria, we defined before the number of rejected hypotheses, let's call them the number of discoveries, the number of false discoveries. This is the joint work of me with Yossi Hochberg, started in 89, appears in 95. Now, we look at the type 1 error in the entire study, not by the number of errors, but the relative number of errors. That is, we look at the proportion of false discovery among the discoveries, V over R, the proportion of them, and put it at 0 if nothing was rejected, and define the false discovery rate to be the expected value of this proportion. That's the difference point of view. Let me discuss for quickly why does it make sense. If I inspect 100 features and we get 2 false ones among 50 discovered, bearable. It's statistics. I mean, we are ready, even if we do one hypothesis, we are ready to bear some error. I mean, we can't, otherwise we will not make any conclusion if we are not ready to bear error. So 2 out of 50 is fine. But 2 false one out of 4, well, that's too much to bear. So in that sense, I do not decide beforehand whether 2 is good or 2 is bad. It is adaptive to the situation I'm facing. The same argument holds when I test 10,000 features. I'm looking at the discoveries, not at the number. So in that sense, it is scalable in the computer science notion of it. And if nothing is real, that is, if all the 100 feature or 10,000 features are all false, then the probability of making even one error is exactly the same as the expected value of this proportion. Because if everything is false, V equals R always. Everything that I reject is in error. So this one takes the value 1 if I reject something and 0 if I don't. Its expectation is exactly the probability that I will make even one error. So I have this weak protection. First line of protection is satisfied, but then I go on beyond that. Because otherwise, since here I have a random variable whose values is less or equal to 1 and can be strictly less than 1, clearly this one, the FDR, is less or equal to this probability. So if I control this one at alpha, I'm giving up on this protection so I can gain more power. So let me summarize. Simultaneous inference should jointly for all parameters. This kind that I presented, selected inference should hold for the selected parameters the same way it holds for each parameter separately. Okay? But on the average over the selected. Okay? So if I have for a single confidence interval, the expected value that it covers or not covers is 0.95, I want it to have on the average over the selected the same kind of thing. And doing each one separately at the level alpha or confidence interval at the level of 0.95 is not enough, because if I look at the expected number of errors divided by the number of tests performed, that is E of V divided by the number of tests performed, this will be less or equal to alpha if I don't do anything. While FDR control assures that I divide this by the number of tests selected, by the number of rejections that I make. And this is known. Now, this statement will repeat itself again and again during my talk. This is hindsight. We didn't have this hindsight when we designed the false discovery rate. In fact, our original motivation was a paper by Zorich, a physician by training, arguing that most research discoveries might be false when using the regular 0.05 level. And this was our motivation he used the term discovery and so on, and this was the term of motivation. In fact, the famous paper by Ioannidis that has made news again and again. How many people are aware of that? Quite a few. This is exactly the same as this one, the same argument in this one as in this Zorich one. Okay. The question is, can we offer a controlling procedure in this case? Well, interestingly, we can. And this is the one that we gave in our work with Yossi Hochberg. In my work with Yossi Hochberg is the BH, now called BH linear step-up procedure, we called it. And again, we sort the p-values as we did for the whole procedure, from the smallest to the largest. And we take k to be the maximum I, such the ith p-value is less or equal to I over m times q. We stop at the largest one such thing, and we reject all hypotheses. Sometimes mathematics is too complicated. The formula is a formula, but the procedure is very simple and can be done in an Excel spreadsheet. I mean, you order the p-values as we ordered before. You compare the largest one with 0.05 times 17 over 17, looking at the 17 p-value. If this one is less than this one, then fine, but it is not. Okay? So we look at the second one, we compare it to 0.05, 16 over 17, it is not. And how about the 15 over 17 times 0.05? This is still not less than that, and so we continue all the way up, and the first time that this happens is here. We have here 0.0257 and 0.0265. So from here, we don't look at that, we just reject it. Again, another thing that is often overlooked, we don't look at what happens here. Okay? So the fact that this one is bigger than that doesn't matter. Just stop here. Now, see what kind of compromise we got. Remember, this was the value that we did by unadjusted testing. That was the value that we could get by Bonferroni adjustment. What it means that there is a strong indication that there are many true signals here, and therefore we don't have to be that conservative in our appreciation of what is significant and what is not. And we can stop here. That's another graphical way to look at it. We sort the p-values, and the constraint is linear. I over m times 0.05, in this case, is simply the linear line, and we look at the first time that we have a point dropping below the line. A third way to look at it is using adjusted p-value. Since they have a special name in this case, I will put it. We looked at this procedure, right? We compare pi to I over m times q. Well, move the m here and the I here. You have here m times pi divided by I. Look at that. That's really if we reject with pi as a threshold, the expected number of errors we make is m pi. How many did we reject? Well, I, because we're going to reject everything that is smaller than it. Well, that's sort of an intuitive assessment of the false discovery proportion. The expected number of errors divided by the number of it. Well, we greedily just take and make this comparison, take the largest one. And that's equivalent to this one, and that's why we can define the BH-adjusted p-values, and that's called also q-values by Story's terminology, and they are simply the maximum j over all j bigger than I that I look at this thing. And when I do that, I can reject hi if its adjusted p-value is less or equal to the desired clue. So they serve exactly the role of a p-value, and you can import them instead of p-values. So you have the p-values, and you can have an adjusted pH. Now that's important because we will have other methods that offer false discovery rate, so it should come with a statement of how you do it. That's mainly the difference between the q values that are defined like that, but it's important. So how do we control the false discovery rate? That depends on the distribution of the different test statistics and the correlation between them. Remember, we work with the PI, so the distribution of each test statistic by itself doesn't matter. If the test statistics are independent, then the FDR is less, by using this method, the FDR is less or equal to m naught over n times q. M naught is the number of two ones, so this is always less or equal to 1. If they're independent and continuous, there are very surprising results. We have an exact equality. I mean, exact equality here. That's the original result. If they are positive dependent, then again we have inequality. And I'll talk about what is positive dependent in a minute. Everyone has his own positive dependent concept of positive dependent, one of the weakest. And even in the most general way, without any constraint whatsoever, you have the result that the FDR is less or equal to m naught over m times q with 1 over plus a half and so on and so forth, plus 1 over m, which is approximately q log n, plus order. Okay. Now, what is positive dependence? Positive dependence is the idea that if you look at the test statistics, x1 to xm, for any increasing set D, what does it mean increasing set D? Increasing set D is something that if you have a point in D, another point which is bigger than the original one in all of its coordinates is also in D. So this is an increasing set, this is an increasing set, and so on and so forth. It contains, if it contains the points, it contains everything above it. Okay, so for any increasing set, and for any null hypothesis which is true, if I condition on it the value that it gets, the probability that x is in D is increasing in S. So the idea is if this is positive dependence, and you look at this region, but you condition on this one, so you look at actually this probability, then as you move to this one, the probability increases, which is a very natural idea of positive dependency since they go together. Yeah?
So I guess the tests have the form xi greater than some threshold. Right, so there's some assumption about the relationship between the test statistic
And the p-values.
and the p-value. So they
Great.
have to be monotone or like that.
Exactly. That's the next one. Okay, good observation. Yeah, I mean, X is positively dependent in any of the derived senses. Taking comonotone, all increasing or all decreasing transformation in the coordinates will leave the dependency the same. So calculating p-values, just taking the probability under the null of that, and they're all monotone, or taking the probability in the other direction, p-value in another direction. This is also comonotone. So the moving from the test statistics to the p-value is just satisfied. But, and that's why it's important for me, not for two-sided. Once I look at the absolute value of my test statistics, once I look at both positive and negative and want to decide when is it significantly different in either direction, then I run into trouble because they are not, the property doesn't keep itself, the PRDS property. However, we're in good shape because there are important cases covered by PRDS. Multivariate normal with positive correlation is PRDS. Absolute studentized independent normal is PRDS. Even something like you take the PRDS distribution and you divide it by a common denominator, then it's not PRDS, but the theorem still holds. Monotone latent variable, what does monotone latent variable means? It means that you have some underlying variable. It's something that you used, for instance, in testing. You have some underlying ability variable. You don't see it, but it's there. Given its value, the results are independent. So the number of answers that you will answer correctly, once your level of ability is known, will be independent, conditionally. This is also satisfies this PRDS. So many situations in practice do cover it. Important cases not covered by the theory is absolute studentized correlated normal and pairwise comparison. But by practice, it is simulation, partial theoretical result, and so on. And maybe with the hope of arousing your interest, I did include this open problem for two-sided test. For those of you who are interested in this kind of thing. Simulation results show that the worst case when I'm doing two-sided, which is not covered by current theory, when the test statistics have correlation 1. So you have the correlation, the maximum correlation is 1. Asymptotically, we know it holds. I mean, that the worst situation is independent and correlation 1. But this is for finite situation. We don't have that. Under this assumption, the FDR can be analytically worked out and show that the FDR is less or equal to Q times M naught over M as before, times 1 plus, but something that always assures that this is less or equal to Q. So we are still in the situation that it is controlled, but only The question is, can it be proved? It has been waiting now for, I guess, more than 15 years with no success. Yeah.
I lost track. What's M naught here? M is the total number of tests?
M naught is the number of true null hypotheses. M is the number of hypotheses. So this is the proportion of true null hypotheses in my, I don't know it for the time.
Right.
Until now, I don't know it. Next 2 slides, I will try to get a hand on that. Now, this is a very recent result, but still waiting for that. The other one is about pairwise comparisons, and we have all the empirical evidence from simulation, including Tukey's own simulation with Nice, with Jones, and so on. And even though the correlation are both positive and negative, we are quite sure that this is the situation. And there is some theoretical results by Icutielli that if you do each one separately, it does control. I mean, it's again in the air, but we don't have a clear proof. I certainly don't hesitate in using it, either for the two-sided, normally distributed, or for the pairwise. Now, look again at this one. We always have this factor. In all of this, we have this factor. This is a factor of conservativeness, right? I mean, because this is always less or equal to 1. And in fact, it can be much less than 1. So if we could put our hand on that, then we would have a much better procedure, okay? Because if M naught was known, we could use exactly the same procedure, but just increase it by M over M naught, or equivalently work with Qi over M naught rather than M, and we will have exact, or we will use FDR oracle and have exact control of the false discovery rate. And this is the essence of the adaptive procedure, try to estimate M naught, or is it sometimes called pi naught or P naught, from the entire ensemble of P values. And there's a whole industry going on. There are dozens of papers offering new methods. There are review papers, at least 4 or 5 review papers that review the different suggestions how to do it. I would like to point at an interesting one, the graphical approach of Schroeder and Pjotvold, that looked at this one, this kind of plot that we saw before, and said, wait a minute, the large p-values, if they come from true null hypothesis, they have to have a uniform 0-1 distribution, and they should look like the order statistics from a uniform distribution. Well, the order statistic from uniform distribution should lie on a line. Well, let's fit by eye a line like that, and get where it cuts here, and get from here the estimate of M naught. And it's working. It's working nicely, and we have been able to prove, but I'm not going to talk about that because I want to emphasize 3 options that have proven FDR control. Okay, there are very few with proven FDR control. First one is 2-stage procedure, and it goes like that. We will use the BH procedure just a little bit more conservatively. Instead of a level Q, at a level Q over 1 plus Q. So just lower it a little bit. Okay? And then we would use the number rejected in order to estimate the number of true ones by M minus the number rejected. Okay? So if I don't reject anything here, that's the first line of defense, I stop, but otherwise I estimate it by M minus R1. I still have to be a little bit more conservative than that. I multiply that by 1 plus Q. So just a little bit more. Because remember, here in the number rejected, there can hide a few true null hypotheses. I'm allowing Q, proportion Q of true null hypothesis to hide here. So, and then use it again with Q star equals Q M naught over M, and it has proven FDR control under independence. What it means is that I will do it again, but now a little bit more conservative, and I will stop here after 8. Then I'll do it again and walk from here. And now I stop for the first time at a 0.063. Now that's wondrous. I did even better than the 0.05. But there's no limitation to that. I can decide that I don't want that and I want to stop at the 0.05. Because if something is taken out. But the point is that in terms of FDR, that's fine. The second option is the one originally suggested by Story and then modified by Story, Taylor, and Zygmunt. Take some predetermined lambda, say lambda equal a half, and again use the fact that the big p-values give you information about how many true null hypotheses you have. So simply count how many big ones you have, number of pi bigger than this lambda, okay, and take m minus 1 minus that. Sorry. That's the other way around. This should be less or equal because m plus 1 minus that should be the number of large p-values, number of p-values above half plus 1 divided by that, and you have an estimator of how many you have over the entire line from 0 to 1 rather than only from lambda to 1. Correct this. Okay. And also an additional condition that pi has to be less or equal to lambda. If it's half, it's not a problem. And it also has proven FDR control under independence. And some asymptotic control for weak dependency as well. But the weak dependency is weak. In fact, while this method is the most powerful under independent of all the ones that are proven to hold it, it fails to control the FDR for all PRDS. And it's interesting to understand why is that? I'm not sure if I kept it, but the point is that it's not by little, but the FDR may double. Just to give an idea, here I have that the estimated number is 6, and I can go all the way to 0.07. This will be the first one, so I really can gain a lot by this adaptive procedure. But of course, here there is a strong, those are the same animals. There is a strong positive dependency between an animal that lingers a long time, the distance traveled is less. I mean, clearly there's correlation because it's the measured behavior of the same animals. Okay.
Yoav, so we should, is this a good time to stop? It's coffee break time.
Okay,
Would you like to stop? Maybe you have a couple
I will stop in a minute and
questions,
then,
break
yeah,
and
we'll
start
stop in a
again.
minute. So I'll speak why dependency matter. It matters because under correlation, if I look at the first two stages, you see that the distribution of M naught is small. I mean, the spread is small. Here we have 48. This is the true M naught. And I'm conservatively above that, but not too big. This is the two-stage. This is the Story, Zygmunt et al. That has a wider distribution, but it's still fine. But if I'm going to correlation of 0.05, look at the spread of this. And too many times it is negative, and when it's positive, it averages out, but not enough because of the spread, because of the variance is positively dependent. In fact, the worst thing is you have here originally 64 hypotheses being tested, and unfortunately when people see that the estimator is 80 or 90, they say, well, that can't be. Okay, I mean, I have only 64 ones. How can an estimator of M not be 80? So they put it down to 64. Now doing that clearly destroys completely the properties of this estimator. Okay, so I will stop here as suggested, and the next one will continue.